So George, we've just been talking about high temperatures being dangerous for humans and lots of other organisms. What goes wrong? Why can't the organism function just fine at a higher temperature?
Mm-hmm. That's a question that a lot of people have looked at, and in the process of investigating it, they've examined quite a variety of physiological and biochemical systems. If there's a single weak link in the chain in terms of thermal sensitivity, it's probably the membranes that surround our cells or surround the organelles within the cell. There's a universal response of membranes to temperature that's been seen in all types of life, and that is that as you raise the temperature, the membranes become a little looser in structure. They become what's called more fluid, and that reflects the fact that as you warm up a membrane, warm up an organism, the membranes that are roughly half lipid, the lipids start to get relatively fluid and melt. And so what organisms...
So it's like melting soap almost.
It's like melting soap or like melting butter. And when I'm teaching on membrane temperature interactions, what I like to use as an analogy is going from butter to olive oil. Mm-hmm. If you're a cold water fish in Antarctica or in the Arctic, your membranes are more likely to have an olive oil type of lipid. If you're a mammal, you want to have a lipid that's not going to melt at 37 degrees. So every type of organism that's been studied is able to change the types of lipids that it uses. So its membrane isn't too fluid or isn't too rigid. And so cold temperatures can make a membrane too rigid, warm temperatures can make it too fluid. So organisms adjust the lipid composition of their membranes.
But what temperature is good for an organism depends on the temperature it's evolved in. Like those fish that die from too much heat, just above freezing.
That's right. Yeah, the evolved differences in organisms that have spent thousands of millions of years at different temperatures, you see it throughout their biochemistry. We see it, as I mentioned, in the membranes. The membranes have to have this right balance between...
So what's a membrane exactly?
Well, membrane is really the wrapping around the cell. That's called the plasma membrane. But within the cell, there are very intricate networks of membranes as well. But they all have the same basic response to temperature. Heat up the membranes, they get a little bit too loose and fluidy to be
so things start leaking.
But the proteins of organisms also reflect a very strong imprint of adaptation temperature. The stabilities of proteins, like the stabilities of membranes, there has to be a certain trade-off between being so rigid that temperature can't hurt you versus being too fluid so you can't function well. So
here's the concept of trade-offs, right? It's the center of all of this.
Yeah, and the nucleic acids, proteins, and lipids, which are the primary constituents of organisms in terms of large molecules, all show this response to temperature. As temperature goes up, their structures become looser and more fluid. I'm trying to simulate that with my hand here. And during evolution, the proteins change their amino acid sequences in a way that leads to the proper balance between stability and flexibility.
So they become just the right amount of flexibility at the temperature that
That's correct. That's correct. In fact, I think you could take a sample of tissue from an organism. Someone were to hand you a sample of tissue, you had no idea where it was from. You could purify a protein, measure its thermally related properties and say, okay, this is probably a 20-degree organism.
Well, that's kind of cool. Could you then do that with DNA from extinct organisms and make a prediction about what temperature they lived in?
You know, that's a good point. And I think I'm an optimist here. I think you could, because if you have the DNA sequence and you can identify a certain gene, then you can extend your analysis into the amino acid composition of the protein.
You can resynthesize the protein and see what it does.
Exactly. Yeah. So you could do that. I don't think anyone has tried to apply that approach in working with ancient DNA, but you could.
It'd be fun.
Yeah. Yeah. It'd be fun to do it with some of.
Even mastodons, you know.
I was going to say mastodons and mammoths.
Because they're fresh enough DNA.
That exactly. And they presumably had body temperatures, near ours, 37.
With them, you still get proteins, actually, from the frozen versions.
Yeah, well, that's true, too. Yeah, you wouldn't have to do it with DNA, but yeah.
Oh,
but anyway, the important point is there is a very strong imprint of the evolutionary thermal history of organisms in all of their molecules.
Let's go on in a minute and talk about fever.